Hello, my name is Kyla Schmitt, and today I'll be taking you through my summer 2023 surf project funded by CURE and supported through the Dave Sutherland Lab. So in general, the Sutherland Lab likes to focus on studying ocean and ice interactions, specifically in areas where there are glaciers, so they do a lot of work in Alaska and Greenland, but they also study water sediment properties in Coos Bay. And I wanted to kind of marry my involvement in this lab with my personal interest in the more biological side of environmental science, looking at things like plant and animal life. So with this project, I kind of took Dave's interest in water quality, water parameters, things like that, and used those to assess kind of impacts on a biological community. In this case, it was native eelgrass or Zostera marina. So to kind of start off, Zostera marina is a seagrass that's native to the southern Oregon coast, and it really supports and sustains many marine and estuarine ecosystems. So, Zostera marina meets the qualification for what is a keystone species. So if you were to remove that species from the ecosystem would kind of degrade and collapse because it simply provides so many resources and habitat for marine animals. You can see that in figure 1. There's a jellyfish kind of resting amidst a cluster of Zostera marina plants. And it also, due to its roots and its mass, it reduces bank and shoreline erosion, keeping all that sand held in place. Beach regression has been a major issue across many nations and many coastal communities. And so having those organisms there to hold that sand and that coastline sediment in place is super important. And it can also help regulate water parameters, which is kind of interlinked with what I studied this summer. So for all these reasons and more, Zostera marina is a really vital asset for us as conservationists and community members to protect and conserve. And in recent years, it's been experiencing a lot of environmental stressors. This comes from different sources of environmental degradation, primarily from global warming and climate change impacts. So as sea temperatures rise, these Zostera marina communities become stressed from the higher temperatures, which are not within their optimal range of survival. And also things like kind of human accelerated erosion that cause sediment to be present in the channel at higher levels than would normally exist can block out sunlight and further stress these plants. And that can result in a condition called eelgrass wasting disease, which essentially causes the plant to kind of disintegrate and kind of melt away. And this obviously has awful ramifications on the broader ecosystem since they are a keystone species. It removes habitat and food for many marine animals. It can accelerate erosion and thus accelerate these other impacts on wasting disease, as well as not removing those ecosystem services in terms of maintaining water parameters. So I guess the way that the Sutherland Lab kind of ties into this is that Dave was previously involved with a project where he and some other scientists analyzed data from the South Slough National Estuarine Research Reserve. I'll be calling the monitoring stations operated through that organization SNRMs for the purposes of this presentation. And they were using that data collected at those stations, which is collected every 15 minutes, just continuously. And so that data has existed for years and is a very regular source of data about water parameters in Coos Bay. And so they were specifically using water temperature to kind of predict how Zostera marina populations would be sustained temporally. But one concern that kind of Dave had exiting this project was that the accuracy of using that those SNRMs, which are located centrally in the channel, kind of at the deepest point of the channel, may not be super accurate when it comes to predicting the conditions within shallower Zostera marina beds, which tend to be located 1 to 3 meters in depth, more towards the side of the channel rather than the center. And as we know, in this field, water properties can kind of change based on a variety of things, and so you're wondering if the cross-channel dynamics would be significantly different. And so you wanted to sample, what are water parameters looking like on the kind of margins of the channel, and is center channel data a good proxy for what is being experienced in the Zostera marina beds on the edges of those channels where they tend to reside, where their conditions are kind of optimal for them. And so those kind of questions are the basis for this research project. Essentially, this goal of the study was to fill in the gaps in and contextualize the existing water quality data that are collected periodically by these SNRs to improve our understanding of how those data can be used and whether they can be used to kind of look at the estuarian conditions that impact these Zostera marina beds. So in order to achieve that goal, I focused on one primary objective, and I did have a couple of others, which I will explain in a minute, and why I did not place them on this poster. But to the extent of the main goal of this project, the objective was to conduct an audit through a matched pairs test and compare SNRs data with data that I hand collected in the waters close to Z marina bed sites. Other objectives included looking at if a competing invasive species of eelgrass called Zostera japonica was present at the sites, and I did find it present at both of the study sites. And then to look at the types of marine wildlife that are there to kind of reaffirm our idea that these beds provide essential habitat to marine wildlife. And I found, I think, between 10 and 15 species of marine wildlife at each site. This included everything from jellyfish and crabs, fish, to bivalves. So that was a really fun kind of series of surveys to conduct. But the primary goal and objective here were to be looking more at the water conditions. And that is kind of what is most integrated with the Sutherland Lab's previous and future kind of goals and interests. And so that's what I focused on for the purposes of this kind of research that I'm putting forth to the URS. So in order to accomplish these goals and objectives, I needed to develop a methodology. So this research project focused on the area of the Coos Estuary called South Slough, which, as you might guess, runs south from the mouth of the estuary. And within South Slough, I sampled the water parameters at 3 Zostera marina beds near the 2 SMRs, which are located at Charleston Bridge and Valino Island. And I did this during July, August, and September of 2023, using June as a preparation period. So I used a lab multi-parameter meter to collect temperature, pH, salinity in parts per thousand, and total dissolved solids also in parts per thousand, approximately 2.5 centimeters below the water surface. And this is right where kind of the ends of the Zostera marina plant are reaching towards the surface. As you can kind of see in figures 1 and 2, you can see how they kind of drape along the surface. And that part of the plant is a part of the plant that is very susceptible to that eelgrass-based disease. So in addition to collecting this data in the field, I also tried to create context for the broader overview of Zostera marina's kind of positionality within the South Slough. So I used QGIS to map I loaded Google satellite imagery through QGIS to be able to visually see from kind of a bird's eye view where those beds were. And there are some limitations with this method, but I generally just wanted to see what parts of kind of the Coos Estuary channel were habitable to seagrasses in general, and more particularly looking at habitat for Zostera marina beds. So in the end, what I found was that water parameters did vary between hand-collected data at Charleston Bridge and Valino Island, versus that SMERMS data from the central channel. So remember, we're looking at side channel versus center channel. So in July, temperature and pH were higher and salinity was lower in Zostera marina beds than at SMERMS. And in August, different trends appeared with Zostera marina bed parameters being nearly all lower, save for Charleston Bridge salinity, than those at SMERMS. And I'm thinking that salinity is likely explained by a SMERMS equipment malfunction due to its being kind of an outlier in the data set. And so in figure 4, you can see those data points kind of laid out. And so these findings kind of affirm the research question and the hypothesis that potentially center channel SMERMS and data collection are not useful for predicting the specific water conditions as they're experienced in the eelgrass beds. And so that's really important because if we are looking to conserve these Zostera marina beds as the essential resource that they are, then we need to have a better idea of what are the main stressors and problems that they're experiencing. And those things can be kind of identified through the two main proxies of total dissolved solids and temperature, which are really strong influences there on their well-being. And through my kind of geospatial analysis using QGIS, I found that these Zostera marina beds are currently well distributed along the edges of the South Sloop Channel. So as you can see, they aren't really found in the center channel, which is kind of helpful in explaining why it is that these SMERMS collection stations might not quite be as accurate at predicting these water conditions as we might hope. And that makes sense too, because the center channel can be significantly deeper than the margins where the Zostera marina is found. And if you think about there's a lot of complicated water dynamics that occur in estuaries because, you know, it's where fresh water meets seawater, but also we have to think about the tides, we have to think about how the different depths in different parts of the estuary are going to affect temperature, as well as the circulation of fresh water and salt water. And so these are all really complicating factors. And so the best way to really be able to predict Zostera marina conditions is just to go get in there with them and to look at what the conditions are specifically within those beds. In terms of, I guess, predicting locationality, Google satellite imagery is imperfect due to several factors such as surface glare and difficulties differentiating between different seagrass species. And so, as I mentioned earlier, there's the invasive Zostera japonica. There are also some other smaller, like, forms of seaweeds and algae that can form similar-looking masses within the water. However, it does provide a rough blueprint for where the marine and aquatic vegetation tend to reside within the Coos Estuary channel. And so future direction includes taking more samples, being able to better kind of characterize these beds in terms of how extensive they are, how the nativity makeup looks, and do hopefully more work with the other types of data that I collected that aren't seen here, like with the marine wildlife and with the Zostera japonica presence and absence of the sites. And so I look forward to continuing that work, hopefully in the future. Thank you for watching the presentation. And yeah, this was a great project to work on, so I'm glad I had the opportunity to do so.